Good morning to one and all present over here in the series of lectures. Today we have Dr. S. Mahadevakumar with us. My duty is to introduce Mahadevakumar. Dr. S. Mahadevakumar is native of Mysore, Karnataka. Presently, he is working as a scientist B in Forest Pathology Department of KFRI. He obtained his master in botany with first rank from Mysore University with 6 gold medal. With DST Inspire Fellowship in 2011, he obtained PhD degree in botany from the same university. He also awarded CSIR RSIP and worked at Mysore University on rust fungi during 2018 to 2021. Prior to joining KFRI, he was serving as assistant professor of microbiology at Karnataka State Open University for a short period. He also recipient of DST Serve National Postdoctoral Fellowship and Shimoga Agriculture University Postdoctoral Fellowship. He has extensive experience in mycology and plant pathology and primarily focused on the emerging diseases of agro-forestry ecosystem. Currently, he is involved in diagnosis and molecular detection of plant pathogens, particularly fungal and phytoplasma disease associated with forest ecosystem. He has published more than 52 research publications in reputed SCI journals and having 13 book chapters. He has published several new genus and species and new records to science. He is life member of Mycological Society of India, Association of Microbiologists of India, Indian Science Congress Association, and American Phytopathological Society. So with these words, it's my pleasure to welcome our today's speaker to this program.
Very good morning to all of you. At the outset, I thank Dr. Mahadevakumar sir for introducing me and KFRI for giving me this opportunity, and I feel it's privilege and honor. And dear friends, today I'm going to talk about introduction to the plant diseases of agroforestry. Everyone will be knowing, most of us all from science background, and if anyone from the humanities background, they will be knowing plants. And they will be aware of what are plants, what is the difference between healthy plants and diseased plants. It is a common observations we see in and around every day in our life. Okay. So now what is important in this talk, what I'm going to cover here. So important, the disease of agroforestry plants is the one thing. And then how we can classify, how we can identify those plant diseases and what are the steps involved in diagnosis. And important forestries or agroforestry diseases, what we are having. And then future perspectives are how this diagnosis improved from traditional morphocultural methods to molecular techniques. So I will take and an outset how we would like to see the environment in and around and how these plant diseases impact. Everybody wants to see a healthy environment. So you might have visited plantation crop, agriculture crop, or even forestry. If any forest or any plants looks like this, it is not beautiful, it is not scenic, and you may feel not too good to see those scenes, right? You always want to see lush greenish, healthy, more productive plants or crops or systems. Okay, so but when you compare healthy and diseased forests, you will come to know what are the differences, how it will affect your psychology, how it affects the environment, how it affects the society in a greater extent or in a larger context. So now what that means to say, we need to understand plant diseases as a common human, whether it's a science student or a humanities student, or irrespective of that, we should understand the plant diseases. What are these plant diseases? So in a technical terms, if I want to define, these are the changes which are invisible and visible responses in a plant cell or in a tissue caused by an individual, or we call them as a pathogen. Okay, so this is the technical definitions I have drawn. Okay, but how we are going to classify these plant diseases? So everyone will be knowing plant parts, leaves, stems, roots, and other things. If the plant shows wilting symptoms, one can assign that it is associated with some problem. You may not say exactly it is a disease. It could be due to other abiotic factors also. So there are two important factors we need to understand when we are studying of plant diseases. What are the causal agents? Whether it is caused by an biotic factor or it is caused by an abiotic factors. If it is a abiotic factors, we have a different terminologies like disorders and deficiencies. And if it is by a biotic factors, then definitely we can call them as a plant diseases. So you can classify or you can name the diseases based on the symptoms. For example, if the root of the plant is infected, you can call them as root rots. And if the whole plant showing a symptoms, you can compare those two and you can call, and like that spots, if they are appearing on the leaves or blights, or the entire plants will show blights and rust, smudge. So these are the common names you can classify. But again, a different school of people may classify them based on organ in which the disease is associated. For example, root diseases, stem diseases, and foliar diseases. And then crop in a larger context, field crops, forest trees, okay, or vegetable crops, turf crops based on the specificity, plantation crop diseases, orchards, like that. So here I have shown a diversity or different groups of plant diseases. For example, starting from wilt to a rust to a powdery mildew, and then phytoplasma, and then root rot, southern blight, stem blight. A lot of diseases you can see, and then angiosperm parasites and downy mildew, okay, and then the fruit rot. This is a post-harvest diseases, and then on maize cobs, on apple, on plants. So we have a wide variety of diseases. So when we say disease is not a simple one, it's a complex. And then how to organize and how to study them based on the causal organism you are going to classify whether the causal organism is a fungi, bacteria, virus, viroids, okay, spiroplasma, phytoplasma, nematode, okay. So based on the causal organism, we are going to technically classify them, okay. So if we do that, it is very easy to understand and it is very easy to dissect what is the causal organism and to go about further diagnosis, okay. So this is the fungal diseases, bacterial diseases, and then these two are culturable and cellular pathogens, and phytoplasma, viral, and viroid. These three categories are including now newly introduced the spiroplasma, and they all come under subcellular pathogens. So they don't have an individual entity, but they all are always associated with the host plants. Okay, but in case of fungi and bacteria, so they are individual cells, and you can culture them and you can further study them in an artificial medium. But for phytoplasma, viral, and viral diseases, I will come to this individual aspects later with certain examples of phytoplasma diseases, viral diseases, how they are differs, and how to identify all these fungi and bacterial diseases versus difference. Okay, so but nematodes, insect pests, and parasitic plants also cause considerable damage to agroforestry plants and other agro-ecosystem plants and general forestry. You take any of these three ecosystems. Okay, so when we are speaking of productivity, three ecosystems we will consider agro-ecosystem, where all our eatables, food products, everything is produced. And then agroforestry, it is established in a motto to sustain conservation and also to gain more economical benefits. And forestry, natural ecosystem. In all these three, pathogens are going to exist. Okay, so but in agro ecosystem, we always try to control them, because in agro ecosystem, the duration of the croplands is very less, from 3 months, 4 months to an year, or sometimes biennial, like that. So if you are not taking care of that, so ultimately it results in loss of production. Loss of production in agro ecosystem means it ultimately reflects in food security. But in the case of agroforestry plants, again, as I said, the two mottos, one is conservation and the other one is the economical benefits. Okay, if the agroforestry plants are affected, so the first those two aims will be collapsed. You won't do the conservation and you won't get the financial benefits for what you intendedly went for agroforestry establishment. And a natural ecosystem, that is a forestry ecosystem, if the forestry ecosystem is affected with plant diseases, so whatever the benefits we are reaping from the forest ecosystem, it will be reduced. Okay, so how this disease is established in an environment? Okay, what in the previous I said, pathogen is responsible for the disease. Okay, it's true, but whether pathogen alone is responsible, or other factors are not responsible, we need to think that. So in pathology, we have a disease triangle concept where three important factors culminate and results in successful establishment of the disease. One is a pathogen and the other one is environment and host. These three factors should combine together. And what are the conditions? The pathogen will be having two phases, virulent and avirulent nature. Virulent means it has the ability to infect very quickly, fast. Okay, and then environment. So if the environment is suitable and the pathogen is going to successfully establish itself, otherwise it will try to evade or it will try to be removed from the environment. And then host, two factors again, susceptible host and resistance host. If the host is resistant, the pathogen cannot successfully establish the disease. That means it may try to infect, but it will fail to establish the disease. Okay, if the host is susceptible, then it can easily get into the system of the plant. So these three factors are very much essential for an establishment of diseases. This is what in the disease triangle, any one component is missing in this, so we can't see the disease. All three should culminate, should come together, then we can see the establishment of plant diseases. Now let's look into that virtual tour of plant diseases, and I will show you more of images where you will come across. And then one point I would like to stress regarding the plant diseases. Indian forest faces number of problems and they have listed top 10, and out of which plant disease insect pest is one among them. Okay, it is not only in forestry system, in everywhere. How much loss we are facing due to these pathogens, up to 40% production loss. Why we are facing a lot of shortage of agriculture products or farm produce, and the actual quantity of yield expected, we are not reaching every year. It may be due to other reasons, but the primary reason is plant pathogens. Okay, so I would like to take two examples from the history, which showed us how one pathogen can change the entire human civilization. Okay, all of you all will be knowing about Irish famine, or commonly called as the Great Hunger. It has occurred in 1840s, 1847 or 1850s, like that. Okay, so it is caused by one pathogen, that is Phytophthora infestans on potato. Usually in Ireland and even today in US, they go for monoculture. In a huge geographical area, they cultivate one crop. Unlike in our countries, we will maintain diversity of crops. If that in monoculture condition, monocropping system, if the one pathogen comes and it infects, then the entire crop will be infected, because the time duration required for the spread of this disease is very minimal. In matter of 24 hours or 48 hours, it can travel kilometers together, thousands of kilometers. Okay, so this Great Hunger disease or this Irish famine left a message that one pathogen can change the social impact. For example, 1 million people died due to hunger, because whatever the potato they grown is completely vanished, and more than 2 million people they migrated to other countries. So that means the social structure is changed. One pathogen, and that pathogen also responsible for the establishment of plant pathology discipline. Earlier we were not having plant pathology discipline. Every life science candidates, they were studying the plants, animals in a nutshell. But now, because of this impact on human life, they established plant pathology. One more example I will take. I will not go for a Bengal famine because it involves socio-political issues also. But how this coffee rust? Everyone will be knowing coffee plantations in most of the agroforestry ecosystem. But how this coffee rust is important in the history of plant pathology. In general term, if I say one pathogen responsible for change of economy and change of economy of one country and change of drinking habit of another country. You think that all this coffee grown in Ceylon, they were used by British people. And once the coffee is vanished due to coffee rust, they switched over to tea. Okay, so this one disease changed the habit and also the economy of Similis or Ceylon or Sri Lankans. Okay, it's caused by Hemilia vastatrix, and the two important messages or important landmarks in the history of plant pathology, which tells us how important these plant pathogens are for our life. If we ignore these plant pathogens, we may come up, or in the end, we may lose all our produce. We may not get anything. Okay, other than these major impacts, they may add toxins to our food. So most of the plant pathogens, they produce a lot of toxins. So this indirectly or directly affects our human civilization or humans directly, human health. Okay, so these are the typical agroforestry ecosystem we are going to study. It could be various timber yielding plants, fruit yielding plants, plantation crops, and others. The only thing is forestry is integrated in the agriculture ecosystem. Okay, some of the major diseases what we have seen in agroforestry ecosystems are starting from early phase. When we take the entire duration from early phase, like from nursery to transferring to the field. So in nursery, we will find root rot, stem blight, okay, and other diseases, wilts like that. But once we transfer them into the field, so if they successfully establish, so continuously there will be a fight between the host and pathogens. But so whenever the host becomes susceptible, so most common diseases we come across is dieback, stem blight, and leaf spots. These are the common pathogens. So these are the, and recent one outbreak I am going to take from this report from Brazil. Okay, so this is on Tectona grandis, and caused by Transilla zonata from Brazil. So we have seen many fungal pathogens on Tectona from Indian context also, or from the southern India. So there are many pathogens at the nursery or also on farm stations, okay, like Fusarium solani, Rhizoctania, and other some Ganoderma diseases associated with. But we are more ignorant about new emerging pathogens. Whether we are strictly scrutinizing what are the new emerging diseases in forestry plants or in the agroforestry plants or in the agro ecosystem, we should be vigilant, and we should every time we need to follow up whether new pathogen is entered or the old pathogens are there. Is there any change in population structures like that? Okay, so another one, again on Tectona caused by Felinus noxillus. Again, it is a mushroom which is causing decaying of the teakuts. Okay, so if this, so mushrooms are silent killers. Many a times we may not see the conchs or fruiting bodies, but the mycelium will be invading into the tissue of the tree components. So that will degrade the value as well as the product of those plants which are affected. So these are some of the examples of agroforestry plants caused by Armillaria species. So you can see different fruiting bodies. So these fruiting bodies developed when they got the suitable environments or when we get the rain. But infections or propagules or inoculum will be present in the host tissues. So during unfavorable condition also, they will be there. Once you, even after taking those products, cut the teak hood, and if you take for further processing, then they may also produce these fruiting bodies. That means propagules we are carrying along with the product. These are a few more examples of how these mushrooms are causing problems for agroforestry plants. Okay, and then few more. Why I'm taking these two examples again with respect to teak. All of us know that teak is one of the very good timber yielding plants, and agroforestry plants are an important component of forestry ecosystem. Okay, so why these people are getting all this? Are we vigilant, or whether our ecosystem has got these pathogens? If they come and appears in our systems, whether we are prepared to conquer those or to ward off those pathogens. So we need to be very vigilant, and these are the diseases caused by Staphylococcus basicola from Brazil. So another one, a trunk canker disease caused by Lacidiplodia theobromine. These are the few examples. But ultimately, what we are going to follow a general set of guidelines. We call them as diagnosis. How we are diagnosing plant diseases. Okay, how we are diagnosing diseases of agroforestry plants. Usually, regular field inspections will be there. Okay, if any outcomes, any symptoms, sign we call them as. So if any sign, symptoms of infections are there, we are going to take them and we are going to process in laboratory for isolation and identification. And that isolation and identification involves several steps, different techniques like morphocultural identification and molecular identifications. Whether we are in the early phase or in the late phase, whether we are relying more on morphocultural or whether we need to supplement or we need to bridge morphology with molecular data. Again, I cannot say that you just go by molecular data and you just do only morphology data. We need to accept what are the recent advancement in science and technology. So if I say only morphology data should be reliable, then what is the use of molecular data? So there are several questions that I will address in the end. Okay, another important groups of forest pathogens or agroforestry diseases are rust fungi. Rust fungi are the one group of organisms which exhibits 5 different spore stages. I think in nowhere else you will find the occurrence of 5 different spore stages in a pathogen or in a microorganisms. So they are ichthyospores, pycnospores, teleospores. So based on that occurrence of the spores, we have 3 different classes of rust fungi called macrocyclic, microcyclic, and demicyclic. Macrocyclic means all 5 stages you will observe. And in microcyclic, uridinia and telia both will be present. And in demicyclic, iridinyal stage is absent. So, and some macrocyclic rust, which prefers an alternative host. As I said, all 5 spore stages will not occur on the same stage, same plant. So there might be a possibility that few phases will be represented in one host and few phases will be represented in another host. The best example is wheat rust. In wheat rust, they have got iridospores and teleospores in one stage and barbaris and few more stages in the barbaris, and that was discovered very recently in 2002 and 2004 by an US group of researchers. Okay, so that means identifying alternative host, whether they are related and all, it will take a lot of time, lot of research investment, lot of research hypothesis. So these are the few rust symptoms what we have collected. This all from my personal collections. This is the Puccinia mysorensis we reported from a wild coffee that is called Psychotria nervosa. Okay, and the general disease symptoms of rust fungi. So what is the impact of these rust in general? As I said, similis rust fungi or coffee rust, you know the impact of this coffee rust, how it has changed the transformation of coffee plantation to tea plantations. And another best example I can quote is wheat rust. And even in Iran, it's Israel and US, most of the 40% of the GDP is contributed to agriculture, and out of that 40%, 90% of the research is focused on wheat rust management. If they lose wheat, they lose most of the staple food. In India, at least, we have a diversity of staple foods. This varies from state to state. But in US and Israel, they're having one maize and wheat. Okay, so they're they're witnessing a lot of losses because of this rust fungi. So we need to be very careful. So different hosts, they're all, why I said alternative host, it could be a wild plant, may not be associated with an economically important plant, but over a period of time, they shift the host plants. So that's why we should be more vigilant. So this disease is on Polyalthia longifolia. You might be aware it is an important component in agro ecosystem or in social forestry or in general avenue trees. Okay, and most of the people will grow as a ornamental plants in office. Okay, outside. So look at the symptoms, how it is colonizing. This is the ichthyospores which occur. All foliage is affected, and then young buds, they are colonized with rust disease. If such disease are present in India, do we have an example? Yes, pine blister, white pine blister caused by Clostridium rubicola. It's a major problem in US as well as in India. Okay, rust fungi, they are going to be a major problem. They are a major problem actually. And this example is sisu. All of you know that Dalbergia sisu. And then rust fungi. Again, we are in the 21st century and still we are dilemma about to accept the old name and the recent taxonomical changes. And so far, these rust fungi, we don't have a cure for this. They are obligate parasites. When we understand the biology of these pathogens, there are 3 important groups we will find: necrotropes, biotropes, and hemibiotropes. Necrotropes are the one which kills the living tissue after colonization. Biotropes, they are not going to kill, but they are going to utilize the host and they draw nutrition and they survive. The best example is rust fungi or biotrophic fungi. Okay, and other downy mildew fungi, they're all biotrophic fungi. But what does it mean by hemibiotropes? So fungi which comes under hemibiotropes, they will show early biotrophism. Later, after successful establishment, they will show necrotrophism. So there is a transition. That's why we call them as hemibiotropes. Okay, so then another important groups of plant pathogens, phytoplasma. So till 1970s, everyone assigning any plant malformations, it could be a phyllodes, it could be a little leaf, or it could be a stunting growth, to a mycoplasma like organisms. Okay, so but in 1967, Doi et al. From Japanese scientists, they distinguished mycoplasma like organisms from phytoplasma. They are very particular. So that's why they coined the term phytoplasma. And these phytoplasma we have a different classification and identification pattern. As I said, phytoplasma, virus, and viroids, they are non-culturable. So if they are non-culturable, it is a serious question for us how to go about research, how to identify them, how to assign them. If I say that I can identify a given phytoplasma based on morphology, then I'm a fool. No, it is not possible. The same symptoms can be exhibited by different phytoplasma. Okay, best example I can show you here is little leaf and virescens. So both are on the same plant. Okay, but if they are occurring on the same field, so we will find a common phytoplasma because we have reported and we have done research on this plant. But if only virescens is associated in one particular region and phyllodes and little leaf or stunting in another regions, they are come up with different phytoplasma groups. So how we are going to identify these phytoplasma groups based on the availability of molecular markers, PCR techniques and nested PCR assays. So this is a chili phytoplasma. Why I am bringing agro ecosystem plants? Okay, so these phytoplasma are transmitted by vectors mainly. They are not transmitted by plant to plant. So vectors play an important role, and they are going to suck from the infected plants and they are going to feed on other healthy plants. And the movement of vectors can determine how impact it could be. Okay, so if it is a peripheral or it will be the geography is restricted, then phytoplasma transmission will be there on that particular region. So if it is having a larger geographical coverage, definitely it is going to carry a long distance. That means one plant inoculum, source of inoculum can spread the inoculum to a wider geographical regions. And again, so the phytoplasma associated with agriculture crops or the wild crops, they serves as a reservoir. Okay, so the same phytoplasma can reside on important tree plantations or an agriculture crop or in a wild crop. So wherever insects feeds on them, they can transmit. So that's why wilderness is more here. Okay, so these are the leafy vegetables, and then this is chili. This is Amaranthus spinosus infected with phytoplasma. Again, this is Callistophus chinensis, is a flowering crop, and as I said in the previous case, all these are caused by one phytoplasma. But you look at the symptoms. So same plant phytoplasma, but yellow, astrellos and virescens, different phytoplasma. Okay. And then this is a flower of chrysanthemum. Due to malformation or due to phytoplasma infections, okay, everything is transformed. See, look at this bunch. Okay, we will not see the florets. Instead, it becomes a greenish structures. Okay, so it may looks like a tomato plant for you, but closer observation, you look at these symptoms, the tip. Okay, and one more closer look of this. This is big bud, right? So once phytoplasmas are infected to an agriculture crop or an agro-forestry plants or a forestry plants, the plant growth will be reduced. Whatever you are expecting from a plant, the product or economic benefits will be declined or reduced to a greater extent. So look at this. This is at the flowering stage. Okay. This is before flower formation. Okay. This whole plant becomes dead. And if the phytoplasma infects at the flowering stage, then fruit will be affected. Okay, so and then in both stages you can see, look at that. Even if this part produces flowers, it won't be in a normal shape and size. Okay, and then best example for forestry or agroforestry plant, eucalyptus phytoplasma. Okay, so this one plant is enough. This one plant is enough to carry the inoculum. Because these tree species are perennial. If the inoculum is transferred from one plant to another, it is not necessary it should produce the symptoms in next 15 days. It may produce the symptoms in next year, next 6 months, or next 2 years. Okay, it depends on successful establishment of those inoculum. Okay, so bamboo phytoplasma. Bamboo is one of the important agroforestry ecosystem, in Kerala and in other southern Karnataka. So look at this cassia, malformations or phyllodes, fasciations, all these symptoms. And then again, one tree, wild plants. So these are all caused by phytoplasma. As I said, there is no cultural methods to identify them. We need to rely on molecular techniques. That means a well-sophisticated laboratory. If we don't have that sophisticated laboratory and sequence technology or service accessible to us, then it may be difficult for us to identify what is the phytoplasma and how to identify them. Okay. So then again, it is a wild plant, Leonidas nepatifolium. All are acts as a reservoirs. If the phytoplasmas are not associated with the wild plants, then we would have saved other important plants. It could be um, commercial crop, or it could be a tree crop, or other forestry plants. So look at this diversity of symptoms. This is Tinospora, Parthenium, and Croton. Okay, and again, Jujubes. There are many plants. The diversity, expression symptoms. Okay, but whatever the plants and photographs have shown, okay, they are representing different phytoplasma. I am not specifying what groups and all, because it may not be beneficial for you people, but you must be knowing what are phytoplasma, how the symptoms, diversity of symptoms they can exhibit, and how the impact. Okay, these are the wild solanum species. Okay, we are comparing with the brinjal phytoplasma, and they are exactly identical, and the vectors involved in possible transmission of this phytoplasma, they are similar compared to that. Okay, from traditional diagnosis to molecular diagnosis, as I said in the beginning, okay, we need to rely on for identification and diagnosis of viral, viroid, and phytoplasma diseases. So molecular tools are very much essential. And again, what about the case for fungi and bacteria? For bacteria and fungi, can we rely on morphology? No. So what is the recent developments in this? So when you isolate a culture, so we are going to identify them based on spore morphology, cultural characteristics, and all. But many of them are showing cryptic behavior. So morphologically, they look similar. And even to that extent, the initial markers used in molecular identification, like ITS, they also exhibit cryptic, and they are not resolving the identity of the given fungal species. For example, best example is Fusarium, Colatotrichum. So all these, they make a complex organisms, complex group. So many of us who are studied traditional taxonomy, traditional morphocultural studies, we will be able to identify Colatotrichum gliosporid is very simple based on spore morphology. But now, based on molecular data, it clearly distinguishes that there are many species within that Colatotrichum gliosporid is complex. Okay, so how we go, that's why molecular tools are very much essential to identify and to designate a particular species trait. Okay, what are the steps involved and all I have shown. Okay, management of plant diseases. Whatever we do diagnosis, we do identify them, we will give a name for this. Okay, that will not end the story. Only thing is, once we come up with the management practices, then only we will be able to successfully control that. Rather, I don't want to say that we are going to eradicate to a certain extent. So in some cases, eradication is must essential, but we can manage. How we can manage plant diseases? There are 6 principles: avoidance, exclusion, eradication, protection, development of host resistance, and the therapy. Okay, individual, they have their own limitations and advantages. Avoidance. Say, for example, if you are introducing a new plant in a new agro ecosystem or agroforestry ecosystem, we can properly screen them whether it carries any pathogen or not. So by that way, we can avoid the entry of a pathogen. Okay, exclusion. If any pathogen is already newly establishing, if any pathogen is newly establishing in an ecosystem, then we can think of that 1 or 2 plants can be removed. That too I am specifying before successful establishment. If it is successfully established, you cannot completely remove the whole ecosystem, right? So then eradication. For example, in certain cases of phytoplasma virus, if you find the first inoculum, you uproot and you burn it. Okay, otherwise they are going to spread by vectors. Protection. If the plants are very important and no way we cannot remove it and we cannot avoid, it is already established, we can take help of chemicals, okay, other protectants. Okay, so based on the type of pathogens you are trying. So for that, a diagnosis is very much important. For if without diagnosis, if you are applying, because in most of the agricultural field we have seen, without knowing what is the problem, they will be spraying only insecticide. When we go for sample collections, we will see presence of fungi, bacteria, but for the treatment, they will be spraying pesticides. There is no connections at all. That means they are not taking proper advice. They are not diagnosing their problem for their plants. So without doing so, simply they are adding chemicals to that field. So that is going to disturb the soil ecosystem, soil fertility, and other species. Okay. And then development of host resistance. So usually in natural ecosystem, plants will try to evade the infections by pathogens. Okay, so we need to find which is the resistant one, and we need to pick them, and we need to subject it for breeding program, so that we can get resistant plants. If we get the resistant plants, then it is easy. Even if the inoculum is present, we can successfully evade that. Okay, otherwise it is difficult. So just think of high commercial value trees. So if it is not applicable for weeds or some small scale plant industries, say for example Mayur phytoplasma. Okay, recently in the Malayalam newspaper, it is there. It is they're trying to cut down those trees due to phytoplasma, and you know the value of phytoplasma and sandal trees. Okay, and it is caused by that phytoplasma. If the phytoplasma is spreading like anything, and definitely they are going to face the loss. Okay, if they want to protect, they should get correct diagnosis. Without correct diagnosis, even if they cut, inoculum will be there and insect populations will be there. They are going to affect the other alternative host. From alternative host, they are going to shift to again normal host ranges. Okay, what apart from this, what are the other methods of management of plant diseases? So the best for example, in agroforestry and forestry, we cannot go for spraying of fungicides, spraying of antibiotics, spraying of pesticides. Usually it is not recommended unless otherwise it is warranted. It is a serious problem. Okay, so biocontrol agents are very much important in these systems. So we can start providing biocontrol agents at the nursery stage itself, so that apart from suppressing the inoculum of plant pathogens, so these bioinoculants or biocontrol agents will increase the immunity of plants, so that they will get more strength to invade or evade those pathogens attack during the entire life cycle of the plant. Okay, and then if the rhizosphere is good, if the rhizosphere microflora is good, then the plant will show more resistance. Otherwise, so they are going to be more susceptible in the future. Okay, so these are some of the photos what we have done in vitro lab conditions for antagonism. This principle is going to apply in nature, in root, in rhizosphere. Biocontrol agents, they are going to fight with those pathogens if it is a soil borne. Okay, so if it is an airborne, other parts, the plant will be benefited with immune resistance, so that will try to evade those pathogens. Okay, so in conclusion, what I would like to say is, plant diseases are changing. We need to document, and we need to be vigilant to record what are the new things, what are the existing things, whether existing, the old one are the same, okay, or changed, and the new one, what are the new one, how they came, and what are the roots of that infection. We need to think of documentation, preparation of new outbreaks or occurrence of severe pathogens on important trees is very much important. And reinvestigation of previously known fungal diseases, and the appraisal using molecular tools and techniques. This is very much required, because if you want to compete with the other Western countries researchers, okay, we need to report in a higher standard journals. So this is the prerequisites. If I do only morphology, if I do some basics, and if I want to go for publications, they won't accept. So it is required to strengthen our knowledge as well as to get benefits out of it. Okay. So I conclude that if any questions are there,